everybody, thank you for being here. So today we are going to speak about probiotic, prebiotics, postbiotics. So we will review the regulatory part in Europe, the clinical benefits, the scientific requirements for clinical studies, and after vigilance points to develop a product with probiotics. So yeah. I don't know. There is an issue for the to go. Okay. Higher. Okay. So we will go through definitions first to be sure that we know exactly the scientific also requirements for this definition. The first is for probiotics, so it's a live microorganism which, when administered in adequate amounts, will confer a health benefit. So two key concepts here. The first is the viability. So we need to demonstrate the viability of the strain during the whole shelf life of the product, and the efficacy with clinical data on the microorganism. So traditional fermented food will be out of the scope of probiotics because we don't have a specific quantity. Generally, the microorganism is not really well defined, and also we cannot really distinguish between the health benefit of the food matrix and the microorganism. So after we will go through the prebiotics definition. So the definition has been a little bit updated through time, but now it's a substrate that is selectively utilized by a host microorganism to confer health benefits. So three criteria: it should not be absorbed by the gastrointestinal tract. It needs to be selectively fermented or utilized by the gut microbiota, and also it needs to refer to confer health benefits. So generally, it will influence the composition of the gut microbiota or its activity. For postbiotics, it's a preparation of an inanimate microorganism or its component that confers also a health benefits. What is interesting for postbiotics is the stability. It has a higher stability because for live probiotics, generally, it may be sensitive to oxygen, to light, or humidity. But there are some requirements: the characterization of the strain and the justification of the inactivation of the strain, and a full characterization of the procedure and the microorganism as a starting material. And also, of course, the safety data and the health benefits needs to be demonstrated. What is interesting also is the side of action. Postbiotics is not limited to the gut. It can be for the skin, for the nasopharynx, etc. So it's a little bit different from probiotics for this aspect. After we will see the synbiotics. So it's a mixture of a live microorganism and a substrate selectively utilized by the co-administered microorganism. We distinguish two categories. First one is a complementary symbiotic, so it's a classical prebiotic with a probiotic, but there is also a synergistic synbiotics, or a substrate is selectively utilized by a microorganism. But both the substrate is not, it's not mandatory that it meets the definition of a prebiotic. Exactly the same for the microorganism. It don't need to meet the definition of a probiotic, but for both, we need to demonstrate the health benefits. So this is the scientific requirement to meet the official definition. For all for all biotics, we need to demonstrate the safety, the characterization, proper condition of use, and health benefits. What is a little bit specific for prebiotic is really it needs to be utilized by the gut microbiota. And for synergistic synbiotics, the substrate need to be demonstrated to be utilized by the co-administered microorganism. So really, the scientific requirements are a little bit different, but it needs to be justified. The regulatory framework in Europe, so we will distinguish two aspects of the regulatory. The first one is the regulatory review of a formula with probiotics or prebiotics. So, in fact, they are not regulated at EU level. We need to go through each member state with their local guidelines or requirements or regulations. We have the EFSA QPS list, which is the first tool we can use because it's a positive list of bacteria, virus, fungi, yeast extract that is considered safe in foods and generally has an history of use in Europe. So that's the first thing. But the category, when we have a look at a regulatory formula, an analysis of the formula, in fact, the category is not probiotic, it's a microorganism, or for prebiotics, it's fiber, etc. So the regulatory analysis is really based on this regulatory category. We have national guidelines for Italy, for example. They have a guidelines on pro and prebiotics with a minimum quantity of 10 to the power of 9 live cells per day. So it's based on the clinical study. And we have also national guidelines. The French Syndicate of Food Supplements published also national guidelines for probiotics. What is a little bit different is the claim. The claim, as we said, is not harmonized in EU, contains pro or prebiotics is considered as an health claim because the terms pre or probiotics will imply a health benefits. So the use of the terms is considered as general action. So we need to have a specific health claim with the terms pro or prebiotics. But there is currently no authorized health claim. So the term probiotic is not, cannot be justified with a specific health claim. For prebiotic, it's a little bit different because we have a specific authorized health claim. For example, for inulin, we have an authorized health claim for bowel function. So the terms prebiotic can be used with this specific health claim. In fact, in several EU countries, the terms has been authorized. Good news for Italy, for example, the terms probiotics can be used for the category of products and also for health claim. It promotes the intestinal flora balance. Exactly the same for prebiotics. The health claim is authorized. For Czech Republic, they accept the nutritional claim contains probiotics or contains prebiotics. For Netherlands, it's only the category that is authorized, so it's food supplements with probiotics, so it's the legal denomination. Exactly the same for Denmark. And Spain, they use the mutual recognition principle. So if there is a food supplements in Italy with probiotics, registered in Spain, they will use the mutual recognition principle. In France, the term is not authorized. So there is a good dynamic in Europe to harmonize now this regulation on probiotics. So now we will go to the mechanism of action and the main health benefits. The first is, in fact, probiotics will have four types of mechanism of action. The first is immunological effects. So it will increase the production of immunoglobulin A. For example, it will modulate the cytokine profile for an anti-inflammatory profile and don't regulate the pro-inflammatory profile, or it will increase the antigen presentation to B lymphocyte, for example, or it will increase the differentiation of several T cells. So there is really a local immunological effect, for example. There are also digestive, of course, effects. It will enter in competition for the colonization of pathogens, or will reduce the production of toxin of pathogens. Also, there is a really good effect on the intestinal barrier function. So it will increase the turnover of enterocytes. It will reduce the intestinal permeability. So there is obviously an anti-inflammatory action based on this. It will improve the tight junction proteins and promote the mucus layer. So there is really a local intestinal action. And it will also inhibit pathogens, produce antimicrobial peptide, and also there is a direct regulation of the transit, the bowel function, via via the enteric nervous system, and also on the epithelial cell. So there is these two. They are the two main actions. But there is also general action: antioxidant, anti-inflammatory, production of enzyme, lactase, bile salt hydrolyze, and there is also neurological effects. It's less known, but some microorganisms can produce neurotransmitters or neurochemicals with neuromodulatory action, so and have via the gut-brain axis a neurological effect. So now we will see the clinical data we have based mostly with meta-analysis. So the first will be on digestive health. The main clinical benefits are for diarrhea, so it can be antibiotics associated diarrhea. It can be also for the improvement of the immune response, especially for infection disease or respiratory disease, and also all the digestive symptoms for IBS, for IBD, or for I would say the general, also the general healthy population, but with digestive symptoms. So that are the main clinical benefits in digestive health. Also on IBD especially, we have some meta-analysis with really a good action for the induction remission rate for ulcerative colitis especially, and the reduction of the disease activity, and also for IBS. An improvement of the digestive symptoms, pain scores, and all the digestive symptoms, bloating, etc. So we have a good scientific body of evidence for this IBD and IBS, which are really in an increasing number as for all chronic inflammatory disease. So now we will see also for metabolic health, for obesity, diabetes, and also for cardiovascular health. So it's the main, I would say initially it was more on digestive health, and now it moves to metabolic health. So there are several mechanisms of action. The first is a general antioxidant and anti-inflammatory action, of course, but there is also as with probiotics, we will have a higher production of short-chain fatty acids. It can regulate directly lipogenesis and triglyceride accumulation in adipose tissue. It will improve the intestinal barrier and permeability. And there are identified cellular pathways to improve the insulin sensitivity and fatty acid oxidation and the differentiation of adipogenesis. So there is really some specific action and also on adipose tissue. And for some, there is also an anti-hypertensive action for some. So now what we can see in several meta-analyses, especially randomized control trials on obese or overweight people, we have an improvement of the lipid profile in several meta-analyses, a reduction of the blood pressure, also an improvement in the glycemic control, so glycated hemoglobin, hyperglycemia, and also all the anthropometric measures, so BMI, body weight, fat mass, and waist circumference. So there is a good body of evidence, but but we need to be careful because there is a minimal duration of use of 1 month and a half or 8 weeks, and also a minimum quantity in meta-analysis, so 10 to the power of 10 or 10 to the power of 9 at minimum. So it's really dependent on the dosing and the duration. So now we will go to the cognitive health. So it's a little bit new, and there is a little bit less of clinical studies, but it's a growing body of evidence, so it needs to be also, I would say, needs to be also discussed. So the first is just a reminder on the gut-brain axis. So the gut is able to communicate with the intestine, with the brain. So we have several pathways. The first is a big directional communication from the neural pathway, because in fact the autonomic system, both the parasympathetic and the sympathetic, has afferent signal from the intestine to the central nervous system, and also from the central nervous system to the intestine via the vagal nerve and also the enteric nervous system. We have also a second pathway, which is the immune pathway. So the intestine is able to produce some cytokines that we circulate in the blood and go to the brain. And also we have the endocrine pathway with the production of ACTH and cortisol, which modulate also the intestinal function. So now we will see a little bit of some clinical data on it. The first is some probiotics will normalize some anxiety or depression-like symptoms. And also for Alzheimer's disease, for mild cognitive impairments, we have an improvement of the cognitive function. It's strain-specific. So, but there is a growing body of evidence based on this. Now we will see more we will see now the vigilance point to develop a product, because we see the definitions, the regulatory part, the main clinical benefits. So the first thing is the characterization of the strain. It is mandatory to have this complete name with genus, species, subspecies, a phenotypic characterization, and a safety assessment. The safety assessment, we have guidelines from EFSA to characterize microorganisms. So we need to have a look with whole genome sequencing with antimicrobial drug resistance to be sure that we don't identify some gene responsible for the antibioresistance. We need to have a look at the metabolic activities, the side effects in clinical studies, the toxin production, and also the absence of infectivity in animal model. So it's really important to have this characterization and safety before any efficacy data. So obviously, as we said, we need to have a look at the regulatory status. So is it present in the QPS list? Do we have an history of use before 1997 in Europe? Does the strain is the strain present in the novel food catalog, the union list of novel food, or is there any opinion in the consultation process? So the novel food risk is really to be assessed before any placing to the market in Europe. And after, there is especially for probiotics when the cells are alive, is the viability of the strain needs to be demonstrated during the whole shelf life, and the quantity put on the label needs to be the quantity at the end of the shelf life. So we need to guarantee a minimum quantity. Also, the compatibility. When we formulate with probiotics, the main issue is the compatibility with other ingredients. So we need to have some contact tests for at least 7 days to be sure that there won't be any decrease in the viability of the cell. So we need to count for the cell at the beginning and at the end of the test with 2 groups, probiotics alone and probiotics with the ingredients. The stability of the formula is really important. So there are some optimized process or storage conditions. So aluminum bag, spray freeze drying for sensitive strain to heat, and also dehydrating bottle with some dehydrating layer. Or we can also have a micro encapsulation or enteric capsule. They will improve the viability of the cell after the gastric step. So it's really important to have this delivery system issue, and the stability and viability of the strain is really important. And that's, I would say, the main issue in terms of product development. When we go to, as we said, we need to have a health benefit that is demonstrated on probiotics, on prebiotics, or synbiotics. When we go with clinical studies, there are some specificities to be sure to be compliant with probiotics especially. Generally, we do a randomized double-blind placebo control because it's a gold standard. And it's especially important when we want to measure metabolic health or glycemic control, adiposity. Generally, we do in randomized placebo control trial. There is a specific also an inclusion criteria based on the cessation of antibiotic use, 4 weeks minimum. It's better several months, but we need to be sure that they didn't consume antibiotics before the inclusion in the study. The food supplement use and the medication needs to be addressed during the whole study. And also the diet. We need to monitor the diet because they are source of probiotics or prebiotics, and also because it can be a confounding factors for the outcome. And generally, we have a health outcome and we have a microbiota outcome when we measure the difference in composition or the metabolites, but we need to have also a specific health outcome. And for complementary symbiotics, generally, we have a control group and we have the symbiotics. But for synergetic symbiotics, we need to have 4 parallel arms: control, microorganism alone, the substrate alone, and substrate plus probiotics group. So it's a little bit more complex for the synergetic symbiotics. And also there is some guidelines on when depending on the claim we aim, the specific scientific requirements. So I would say this, just after. The first is also that EFSA gives some guidelines for the outcome. They give, we need to have a specific health outcome, especially for immune system. It will be the severity, the duration of symptoms, or it can be also the severity of digestive symptoms or the frequency. But it cannot be a general outcome on inflammation, on immune cells, etc. So we really need to have a health outcome and after the mechanism of action. If you want a claim contains probiotics, for example, we need to have a proof of viability because we need to demonstrate that the cells are alive during the whole shelf life, as we said. But we need to have also some clinical studies for the health benefits, but it can be on the taxonomical category. For example, for bifidobacteria, it's not strain specific. If we want to claim contains probiotic, for example. If you want to have a specific health claim, I would say helps to reinforce the natural defense of the organism, for example, or for immune system or whatever. We need to have clinical data on the strain specific. because we need also to justify the dose and the duration, the condition of use. And also, if it's probiotics, also to have the viability demonstration. So all depends on the claim that we aim, the scientific requirements. So there is a huge opportunity for probiotics or synbiotics in functional food, in food supplements, in drugs, and in FSMP, donc food for specific medical purposes also. And for the target population, it can be for digestive health, so IBD, IBS, or digestive general comfort. It can be for metabolic health, for obese or overweight or diabetic people, and also for sport nutrition, because as we target the immune regulation, also the inflammation, the oxidative stress, so there is also a huge impact for sports, the aging population, and the general population. When we consume antibiotics for immune system, for digestive comfort or respiratory infection, also for children, especially for digestive comfort. So I would say that there are many target population and also product application. So as a summary, we could say that the first step will be to characterize and to have safety data, and also to have a look at the regulatory status, especially of the strain with the novel food risk. We have the formulation. So do we have a compatibility with the ingredient and the strain? Do we have a demonstration of viability, of stability? And do we have a good storage condition? The efficacy data, so to aim with a good population, to really justify the condition of use, the dose, the duration, and to demonstrate the health benefits. And also generally we have the product notification depending on the product type, category. And generally we can also do some technical dossier for the safety data, the efficacy data, and all the specification. So we would say that we have several steps in the product development. As a conclusion, I will just present RNI, our consulting agency. So we do regulatory affairs, so it can be product registration, regulatory analysis for formula or labeling, novel food dossier, GRAS or NDI to really authorize some ingredients. We have an export strategy. We can do also quality control, and we have a medical and scientific department for the safety and toxicological risk, for clinical studies, for product formulation, and strategy research plans. So our booth is on the N170. So we will be pleased to see you if you have any questions on probiotics or other issue. Thank you for your attention.
Thank you, Veronique, for that very concise presentation. It covered a lot of bases. Thank
you.
You did. And there's lots in there. Any questions for Veronique?
Yeah.
No questions. Oh. Sorry.
What does absence of infectivity mean?
I didn't
absence of inactivity?
Infectivity, yes. In fact, it's for it can carry some infectious. Can I would say trigger some infection. So we need to be sure that it won't trigger some infection in the animal studies generally. Just to be sure that it doesn't carry infectivity. Yes, infection risk.
Absence of pathogenicity.
Yeah, exactly. That it won't trigger an immune response and to be considered as a pathogens.
I have a question for you.
Yeah.
Do you think the regulatory divergence globally will ever align?
I hope so. There is a good dynamic, as we said. There is a good dynamic for harmonization right now, really to say for the type of claims and what is the definition, what is the scientific requirements. What I show you on the scientific requirements is based on expert consensus, so they just need to put the expert consensus in regulation. But yes, in Europe it's really difficult to be harmonized, but there is a good dynamic. So normally it should be harmonized.
A lot of what you presented, IPA was at the table with Health Canada, and we created the probiotic monograph.
Yeah, exactly.
2009, 10, and 11, and that's a model which a lot of governments
Sure.
are looking at right now, and it works because it's a pre-approval process, right? You have to tick all those criteria as you mentioned, and then you apply depending if you want a general claim or a specific claim.
Exactly.
And you have to provide the evidence, and either in 30 days you have a product that goes to market that has a very general claim or something more specific, but you need to prove it with clinicals. So
exactly.
Yeah. And we talked with many governments who want to know more about this model because we do interact. So
Okay.
awesome. Thank you so much.
Thank you.